All right, everybody. Welcome to the Neuroinformatics and Data Sharing session. My name is Alejandro de la Vega, and this is Lei Wang. And I'd like to invite, without any further ado, Dylan Nielson up to the stage. He's going to be presenting Detecting and Harmonizing Scanner and Site Differences in the ABCD study.
Hi, everybody. I'm Dylan Nielson. I'm on the intramural data science and sharing team at the National Institute of Mental Health in America. And so just to clarify for people who aren't familiar, on the intramural side, the NIMH Data Archive and the ABCD folks are separate entities, not associated with them. The things that I say you should not blame them for, blame me only. So today I'm going to be talking about some work that I did trying to detect and harmonize the scanner and site effects in the functional MRI data from the ABCD study. And I'm on the data science and sharing team, but we also had a lot of help from our sister team, the machine learning team, led by Francesco Pereira and Charles Zing, who provided a lot of help with the design and statistical analyses. So the question that started this project, someone came to us and said, we're thinking about how to design really big studies and what the best way to go about this is, and is it better to collect scans on just a single scanner, or should we do it on a whole bunch of scanners, and what do you think some of the trade-offs would be? And I said, that is way above my pay grade, but the question I can answer is, data scientist, my first thought is, okay, how do I not answer that question, but answer one that I think will help them and is something I can actually answer? And so instead, if you collect data on a bunch of scanners, is the information about which scanner it was collected on still present in that data, and are there some things that you can do to get rid of it? So focusing on ABCD for this analysis, it's a great data set, a whole bunch of subjects and sites, a lot of different functional imaging, resting state tasks. In their pre-computed tables, they provide both task-based contrasts and task-based conditions. And I'll be splitting those out in some of the analysis later. And they've done a lot of work to standardize their functional imaging protocols, which you can read about more in that publication. And then we also emailed some exchanges with the ABCD, and we have the full scan cards for the scans that they used available on our GitHub page. And I think they're working on a more permanent way to distribute and share that information. And importantly for me, they released the data as pre-computed tables, so I didn't have to worry about the vagaries of processing data from 3 different manufacturers and a bunch of different models. So it was a great data set to work for this question. And normally, you would use a traveling subject to answer this sort of question, the typical how much variance is accounted for by subject versus scanner in fMRI. A lot of those studies have been done, but that isn't available yet for the ABCD. It may be in the future, but there isn't that data set in the public yet. And so my first approach to answering this question is just, can we classify the scanner that the data was collected on? This is a replication of a study that we published on bioRxiv with the ABC 1.0 subjects, and then we pre-registered on OSF basically a very similar analysis on the ABCD 2.0 subjects. So what I'm going to do is describe the data that we used, look at the scanner classification, describe an approach that we tried to eliminate that scanner variance called COMBAT. It comes from the mini batch arrays in genetics. It stands for combining batches. So it sounds very warlike, but it's not. It's just a geneticist being funny. And then we'll look at some other ways that measuring the percent variance explained with this data, and I'll summarize the results for you. So like I said, lots of subjects, lots of data, lots of sites. In addition to that, there's 151 non-imaging tables, a whole bunch of different imaging modalities. The data that we're looking at is the resting state fMRI. That's about 416 metrics. Those metrics are connectivities between different networks and within different networks and between those networks and a set of subcortical structures. And then we're also looking at the task-based fMRI, which is 3 tasks, in-back, stop signal, and monetary incentive delay as average betas from 98 ROIs from the Deskian Atlas. They provide other atlases. They provide Destro, but we just use the Deskian for this analysis. And importantly, the 4 conditions that they've released in their pre-computed tables are all from in-back, and then stop signal and MID have a couple contrasts, and there's 5 contrasts that are from in-back as well. So one of the really great things about having a whole bunch of data is you can throw a bunch of it away. And so there were a lot of reasons that I wanted to get down to a smaller data set to simplify the analysis. And some of those are, a whole bunch of them I'm just throwing out because they were used in the previous analysis. Taking out the QC aspects, and then just to simplify things for myself, throwing out sites that after all this filtering had fewer than 100 subjects for the resting state or fewer than 50 for the task-based fMRI. Task-based fMRI, that number has gotten so small there at the end because I'm requiring that every subject I use have data for at least one run for 3 tasks. And so that ends up being a fairly strict filtering. So we end up with about 3,500 subjects for the resting state and about 2,000 subjects for the task-based data. Looking at the distributions of gender and just the counts by site, you can see the sites we're using for resting state and task-based data. The gender splits are about 60-40 across all the sites, so there is some variability in that. And you can see we've got 5 GE, 2 Philips, and the rest are Siemens. Looking at the age distributions by gender and modality, you can see, so just to kind of orient you guys, I've just taken a histogram, colored the density, and kind of looked at it from the top down so you can get an idea of how much those distributions are varying between sites. So after all this filtering, there is a good deal of variability between sites. And the reason I bring this up is because it's going to be important to control for these effects when I'm trying to classify the effects of site and not to be classifying accidentally the effects of age or gender. So now let's take a look at that classification. I just did a really straightforward multinomial logistic regression, nothing fancy. I wasn't trying to develop new techniques to determine what scan your data was collected on. You should know that. We did accuracy average over 3-fold cross-validation and then significance assessed via permutation. We didn't want to just go by, what's the chance rate of classifying these scanners because they're slightly unbalanced samples. We've got the age effects, we've got the gender effects. And so we wanted to assess chance classification with permutation to take those into account. So resting state scanner classification, we've got correcting for sex just by balancing our draws for sex, and we get about 25% accuracy for classifying which scanner it was collected on. Balancing for sex and regressing out age is about the same. And then if we balance for sex and age in 3-month bins, the classifiability drops down a little bit, but we're still above 20%, still well above chance. So there is definitely scanner information in that resting state data, which I think is totally understandable. It comes from 3 different manufacturers. And then the task-based data is a little bit better story. The contrast, since there is that internal subtraction, internal control, those are only classifiable very, very slightly above chance. And the conditions, since they don't have that subtraction, are more classifiable at about 15%, and that is, again, significant. So taking a look at combat correction. So as I said, it's a method that came out of genetics. If you want to talk to me more about it, you can come find me at the poster on Wednesday. But the significance for this was assessed with 10,000 bootstraps for the differences between combat-corrected and non-corrected results. So resting state data, we've got the uncorrected again. You've already seen that part. This column here is after correction with combat, and they were all, the classifiability, the accuracy of the classifiers was all significantly reduced, but still somewhat above chance. For the task-based fMRI, the contrast, there wasn't a lot of information there in the first place. Combat might have reduced a little bit, but that's not a significant change. For the conditions, though, there is a significant change, and it does still leave those ever so slightly, the classifier is ever so slightly able to perform above chance there. So looking at percent variance explained in kind of a different way. So the data is in there. The classifier tells us that. What is contributing to that data? What are the relative magnitudes of how these different things are contributing? So we just had a set of nested models and subtracted variance to get the percent variance explained for each of these factors kind of going up the chain. And again, significance assessed with 1,000 permutations. And here I'm showing the resting state data. In blue is the variance explained with the uncorrected data. And each of these, so there's 416 metrics or so in the resting state data, and that's what's shown in this distribution is the percent variance explained across those metrics for manufacturer, for scanner, and then for age and sex. The dashed line here is significance. So as we saw, it's pretty classifiable. There are a lot of metrics that explain a fair bit of the variance in the, there are a lot of metrics for which manufacturer or scanner explain a fair bit of that variance. It's in the 2 to 10% range. The tail on this is really long. For the task-based conditions, as you'd expect again from the previous results, the classifier results, You have a little bit more percent variance explained in the task-based metrics by manufacturer and scanner than you do in the task-based contrasts. And another important note here is that underneath each of these big blue histograms is a little tiny orange histogram, and that's the percent variance explained in those metrics after combat correction. So it goes way, way down, again, as expected, but for agent sex, it's preserved. So when you run combat, you can say, oh, correct this variance, preserve that variance, and it does a good job of preserving the variance you ask it to preserve. And then as an exploratory analysis, I'd seen some talks from the ABCD folks, and they mentioned the effect of family. And so I just ran a normal ICC type analysis with the resting state data. And here, I don't have agent sex in the bottom. I've got family, because I've got a family grouping variable. And family explains a huge portion of the variance in this data compared to the other effects that we've seen. And again, that's preserved with combat. But doing this different approach, we see similar percent variance explained across these metrics by manufacturer and scanner. And so now to summarize, ABCD again is a fantastic data set. They've got an outstanding commitment to open science. It's been really great to work with their data. And as a bonus, I didn't have to collect it or break my back trying to analyze this data from all these different sources. Definitely check out their online analysis platform called Deep. They've done some really cool stuff with that. I didn't use it for this analysis, but it's a great tool that's out there, and it has built-in to help you control for the effects of scanner or manufacturer. And then to summarize my results, the fMRI data does have some scanner information in it, particularly the resting state data and the task-based conditions. The contrast seems to be pretty much okay. It's similar in magnitude to the percent variance explained by agent sex, and about a fifth to a quarter of the magnitude of the percent variance explained by family. People are largely going to be looking for small effects in the ABCD data, so I think it's probably for a lot of these analyses, particularly with the resting state data, going to be important to do something to control for the effects of scanner manufacturer. Combat does a pretty good job. If I ran Combat, I would still use a mixed effects model to help control that additionally. And again, I'd like to thank everybody who helped, and I'll be at poster 582 on Wednesday for anyone who wants to have a more in-depth chat about this.